All right, let's go ahead and get started. Hello, everyone. Thank you for joining us today for our ALS Webinar Wednesday series. My name is Alex Yao. I'm the marketing coordinator for ALS North America, and I'll be facilitating the webinar today. Just a few helpful notes before we begin. If you're having issues or technical questions about our webinar platform, you can type your message or question using the chat function at the bottom of your screen. You can also select the hand icon to notify me of your status so I can assist you. All questions regarding the webinar material will be answered at the end of our presentation. Our presenter today is Ray Martrano. Mr. Martrano is an environmental chemistry leader and specialist with over 35 years of combined experience in analytical chemistry and environmental engineering. Specific areas of expertise include environmental laboratory management, analytical chemistries and methodologies, sampling, project management, remediation technologies, process engineering, and quality assurance and quality control. Today, Ray will be discussing SARS-CoV-2 testing in wastewater. Thanks for taking the time to deliver this presentation. I'll turn it over to you now, Ray.
All right, thank you very much, Alex. And I appreciate everybody attending today. As Alex described a little bit, I'm a bit of a generalist. So I kind of get involved with a little bit of everything. So this is very much a very interesting new area of study. And hopefully today we can kind of cover this in a way that everybody can get a little bit of a grasp of what we're talking about here whenever we're looking at the SARS virus in wastewater. So let's get started. Obviously, the goals for this presentation, just to give you guys a little bit of an overview of what I'm going to try and do, what I'm going to try and present, is I'm going to start out a little bit and try to weave a story, in a sense, to go over, first of all, the virus, where that comes from a little bit, what viruses are, and again, cover things like, introduction to cells and viruses and how they replicate. That's very important because we use those mechanisms actually to detect specific signatures associated with the virus. Talk a little bit again about the genetic testing and specifically PCR, which is the main tool that is used in the virus monitoring in wastewater. And then we're going to jump a little bit after we do our biology lesson, if you will, into the application of biology to industry. We'll talk a little bit about the sampling, lab work, and some of the considerations needed for this testing. And then we'll talk a little bit about monitoring, what you may or may not want to do with regard to surveillance programs and how you can use the data. And finally, we'll discuss a little bit of pros and cons and limitations of the data that you may obtain during your studies. So a little bit of background to start us out is what is a virus? And I've had a biology class, and I remember my professor who discussed some of this stuff. Specifically, we'll talk about the SARS-CoV-2 virus. Don't get confused when people say they have COVID. COVID is the infection. The SARS-CoV-2 is essentially the form or strain of virus that's currently causing havoc in our world right now. It is a form of the human coronaviruses. The coronavirus actually has been around for quite some time, and it's actually named after the spikes on the surface of the virus. And I'm sure we've probably seen a lot of pictures in the news what that is. Viruses are very different than cells. Cells typically have all the bits and parts needed to replicate, and a virus does not. Viruses are signature kind of ways they're detected is they have what they call a protein coat that usually surrounds either a piece of genetic information, which is either RNA or DNA, but never both. These are slippery, slimy creatures. They can go dormant. They can come back alive, and they're quite a lot like parasites where they need a host to reproduce. And whenever they get into a cell, they attack it differently. That all depends on somebody's specific genetics, which is why we're seeing people have a lot of different kind of reactions to this virus. So you'll see these things behave quite differently amongst people. So one of the first things maybe we want to do is talk a little bit about the cycles whenever people get sick or infected. Two very different things whenever we look at a virus versus a common infection. Infection is a host of a bacteria that might get into us something, for example, that might cause like strep throat, where we get like slime in our throat. A virus is quite different because it's a genetic attack on the cell. And think about it a little bit as if you were inside your house. A snowstorm could come and keep you on the inside of the house, which is what infections do. And viruses kind of get inside your house, get in your kitchen and start making a mess out of things. And the way the differences between the two can be thought about. That's why whenever you get a cold, doctor tells you to go home, just let your body take over. Versus if you get an infection, you'll take an antibiotic. And antibiotics don't work on colds because they don't mess with the genetic stuff. Rather, the antibiotic attacks a certain specific process in the bacteria that's causing an illness in you. So whenever we look at the common curves associated with getting sick, initially we have a time zero where we're exposed to some sort of a microbe, or in this case, a virus. Then we'll go through the incubation period. And this is where there's not enough counts of the virus or the microbes present to cause us any type of concern. We don't even know we've been exposed to anything. The next period is this incubation period, which is right in here. And what's happening there now is the virus or the bacteria is multiplying on us. And then all of a sudden we start getting to the next stage where we start getting a bit sick. We start to feel some sympathy. And then obviously this period right here in the middle of the graph is where we're really getting sick. Symptoms are getting a lot worse. And then commonly at the apex of this, we go into the next phase, which is the final phase, which is convalescent period, which is where like maybe up here is where a fever might break, for example. Our body's figured out the genetic code to destroy the virus, and we start getting well. Commonly somewhere in these areas here is the contagious period with that. If you continue to get sick, obviously that line's going to go up off the screen here, and that's ultimately death whenever we can't control or kill an infection or a virus takes over too much. So these are the common cycles, and these are important because this helps us identify the periods in which our bodies have these organisms in that are possibly shed by the feces, and that's how we detect this in wastewater. So let's talk a little bit about how we want to detect them. And so in order for us to do that, we need to know a little bit about how viruses actually work. And this graphic here is going to help us do that. So the first thing that a virus actually does is it's going to come onto a host cell and it's going to attach itself. Typically, one of these spikes is going to attach to a protein that might be on the outside of a cell. I believe for the COVID virus, what happens, it attaches and it actually injects RNA into the cell and starts disrupting the processes in that cell. A lot of times, again, the penetration will happen. We'll get some sort of genetic signature in there. Then what happens is biosynthesis, which is in this part. Typically, a part of an RNA sequence will go in, and rather than making what the normal cell will make, it's going to start making what the virus wants it to do, the different proteins that are commonly manufactured by an individual cell. They will now assemble copies of that virus, and then eventually it will be released. Commonly, whenever viruses are released from cells, it's a process called lysing, where the cell will rupture, and then a newly formed virus or copies of virus will be ready to go out and reinfect everything, and the process starts all over again. The way we're looking at right now with a lot of vaccines are ways to kind of prevent this guy from attaching onto a cell, and that's where a lot of work is going right now. So let's talk a little bit about some of this genetic material, because this is what we're after whenever we study any type of genetic disease, genomes. And typically we have in cells, in the nucleus of cells, are the DNA structures, which is a double helix of nucleic acids. We have RNA, which is essentially a mirror copy of the DNA. And what this guy does is it actually carries the genetic code and makes proteins. The nucleotides, which are these guys, these are the individual building blocks that actually make the polymers. And they are known as typically adenine, thymine, guanine, and cytosine. And in RNA, it's substituted for uracil. And what happens is in me, you, everything that lives, is these sequences really are our genome. It makes our genes. It tells our cells what type of proteins to make, and it is the, if you will, the fundamental building blocks of life. So two important processes that go on in the cells are obviously the replication, and the other part is the transcription. Replication is where the DNA strand is broke, and it's copied. There's typically two strands that are copied. You got a leading strand and a lagging strand. We'll have commonly a little fragment of RNA, which will attach, and we call that a primer. And the primer can be thought of as like the engine on a train, where it's got maybe no hook on the front of it, but on the back, it's got a hook so that additional cars can go on. Here, we'll see the nucleotides that'll go on down the string, and it'll build the DNA, and it does that in both processes on both sides, leading and lagging. One works in this direction, one works in that direction, until it hits a stop, which, if you will, would be another RNA primer, which might be the caboose where nothing is attached on the tail end, but you do have something where you can link onto the front end. So that's the replication process. Transcription is whenever we actually read the DNA, and part of RNA's job is to read this code. And what happens is these bases, bind to the genetic material and makes a copy. And then this is this transcribed RNA. And if you will, that's the signature that goes into the host part of the cell to make the specific proteins that do the job that we're looking at. So whenever we talk about genetic testing a little bit, really we want to kind of read this code and understand it. And think of it a lot like letters in the alphabet or notes in music. We've all seen and heard, many, many words and many, many songs, but ironically, there are only so many letters in the alphabet, or if you will, there's only so many notes in the music. But it's the differences that make the song difference, the words differences, and slight little changes can certainly change a word by just one letter. And again, we have a lot of things that are similar in these. But a lot of things that are unique. So like if I would say to you, like, oh, say, can you see, which might be a part of a genetic song, we would all recognize very quickly that that's the Star Spangled Banner. But if I said something like, oh, baby, then we could think, oh my gosh, that's in almost every song we hear about somebody that's singing about their lover. So again, commonalities and differences do exist. But what we do need to do is we need a tool to identify and detect the differences in this genetic material. And that guy is PCR, okay? And that stands for polymerase chain reaction, which is what we're gonna talk a little bit more in depth here today. So what is PCR and what is qPCR, which are the two tools commonly used in biology laboratories? They are the primary things that we use, and it is a way to look at a lot of genetic material, and it is used in a ton of different applications. And essentially what it does is it works by replicating signature parts of a DNA or an RNA sequence. And what it does is actually it mimics the cell activity. We put all the ingredients together and we mix it up. And if you will, inside a machine, what we're doing is we're actually copying DNA or an RNA. It is a very rapid and it's a very effective technology, as well as it's also very specific. And the qPCR is actually the quantitative version of PCR. And how we quantitate things is we add a primer or a dye to the genetic material. And there's tricks and ways that happens whenever it binds to the sites we want that may quote unquote turn on like a signal or a flashlight. Others are what they call, I believe, quenchers, and they work by actually breaking a material, and if you will, that allows them to illuminate and be detected. So the PCR technology is essentially copies specifically parts of the DNA or the RNA sequences so that we can see the specific parts of the DNA or RNA being studied. Okay, so how does PCR work? Well, again, it's a little bit of adding the same type of things that we find in cells. One of the first things is the primer. And if you remember back to the other slide I just showed, the primer tags a specific spot of the nucleotide that we would want to copy. And it starts It's a starting location for things to attach, bind, and start moving forward. Polymerase, which are kind of like a basic mix of enzymes and nutrients, which help make this process happen. Think about a little bit almost some of your basic ingredients like flour and water and sugar in your kitchen. The nucleotides are again those unique nucleic acids which will be added to the sequence. And those are the building blocks of DNA, as we already discussed. Buffers, if you will, like kind of we want the oven at the right temperature, we want the right pH, we want the right control, and of course the source DNA or RNA that we would like to copy. The PCR device works by doing things in steps or cycles. First is the breaking of the DNA to expose the acid sequence so that it can be copied by base pairs. The annealing process, where once we expose it, then we will add our primers to specific areas of the genetic material that we want to copy so that we can replicate it. So we have starts and stops, just like the train engine and the caboose of the train. So we copy that, all the cars, if you will, in the middle. And then the extension, which is the building of that new genetic material. Now, once that is done, the device works by just repeating these thermal cycles to make copies of genetic materials. So like at one part, you heat it up a little bit, you cool it down, you cool it a little more, then you heat it back up and you keep going cycle after cycle after cycle. What this does is it allows us to amplify or replicate the genetic areas that we're interested in studying. So for example, if this is something that I'm looking to study here, and this is my target area, during the first cycle of the PCR, I take one copy and I get two. The second time I run the cycle, I get double that and double that and double that. And it keeps going so that in 40 cycles, with one piece of, or strand of DNA or RNA, I can make 100 billion copies, or 2 to the 40 of this. So at that point, whenever we add primers and dyes is whenever we can start seeing this stuff. The qPCR, again, is whenever we put those dyes in there. They do attach onto each replication. There is a certain minimum quantity of cycles, if you will, to where we can replicate it, to where we can see it. An analogy I can use maybe to describe that is thinking that if I was sitting on the end of a football field and somebody on the other side had a flashlight, and if there was one flashlight, I may not be able to see it. But whenever I get 2, 4, 8, 16, somehow at 32, maybe I can start seeing that. So those are like your minimum quantities that you can see. So obviously, the more cycles that are necessary through the PCR, the heating, the cooling, the thermal cycles, the lower the concentrations, because we just didn't have enough starting material. And we can use that information then, comparing that against knowns, so we can get some sort of quantitation on it. And that is really the key of that, is to provide a concentration of genetic material per volume of sample, whether it's a nose swab or it's wastewater or if it's surface swipe. But now we can kind of correlate that a little bit. And again, this technology is used in a lot of different applications. Okay, so with that said, we can look at a graph that is actually, this was provided by our Houston laboratory, of an actual PCR curve. And you can see here, starting out first cycle, second cycle, we don't see a lot of stuff, but during 20 cycles now, we start seeing some responses that are coming out. And again, this is a higher concentration material on this end because it takes less cycles to replicate it to where I can start actually seeing it above a threshold. And out here, the more cycles are actually lower concentrations. So this gives you an idea, if you will, some sort of fluorescence based upon the number of copies that we're seeing here. So now we can jump from our lab finally into wastewater. So this can be applied certainly to wastewater testing. And it's a way, if you will, that we can take fragments that we may find being shed from people through feces that have been infected or are infected in wastewater. The technology has already been applied previously to things like polio. That's been studied. You know, obviously polio, hopefully we've eradicated it. But whenever we look at it, studies definitely be overseas a lot on people looking at the polio virus just to see if it's gone. And this is a really good way to kind of look at that. What it does is the individual will see as they go through that curve, at some point in time, their body is going to start shed this material. As they synthesize it, it goes through this system. And that, again, is where we start to pick this up in wastewater. Samples pretty much can be easily obtained in a non-intrusive manner. I mean, we don't need to knock on anybody's doors. We don't need to stick a swab up anybody's nose. And for the most part, most areas or populations are covered by municipal sewage systems. So we can collect these samples at any point or any part of the sewage system. A lot of times it's done right at the headworks as monitoring, but again, very non-intrusive. And again, the technology is very rapidly available everywhere. We're using it for a lot of different applications, and all we've done now is the geneticists that work on these things is they've built specific primers to attach upon the RNA string that is unique to the COVID virus. So what we do is we attach a train engine and a train caboose right to the areas that are specific to the COVID, and we can multiply that, and that's how we detect it. So moving on, let's say now I want to do some sort of monitoring on this. So one of the things you want to start doing is certainly consider how you want to use this tool for any type of monitoring applications. Obviously, anytime you're going to go in the field or anytime you collect the samples, you really have to identify what are your objectives. What do you want to get? Do I want a presence absence? Do I want to know how much? Do I want to see a change in the system? And then obviously you have to consider, where am I going to sample from on this? Location of your sampling collection points are very important. If you, let's say you want to keep an eye on the senior citizen's house, you might want to collect from a sewage outlet right from that facility before it connects onto the main. You want just some broad sampling, collect it right from the headworks. Obviously, with any type of sampling protocol, representativeness is very important. We want to make sure that whenever we go into the field, that our sample is representative of what we're trying to monitor and what we're trying to study. So things like purging, so we get rid of like dead legs, if you will, or dead volumes. How we want to composite a sample. Do I want to look at a snapshot versus a grab, or do I want to like take a 24-hour composite? And then how long do I really want to collect this sample? And it can all be done, depending upon what you want to do. The other important thing is when to collect the samples. You know, if I was looking and I was going to study a university, and I did that over at Christmas and that university's closed, that would not provide me with very good information at all. So the timing of when to collect your samples is very important. Middle of the night, maybe not so much, because might not see so many people up and about, but maybe in the middle of the day, you can definitely see some varying concentrations. The frequency of sampling is the other important aspect of this. Is, I always tell people that, a single data point does not make a line. If you're like me, I get 3 data points and my data always looks like a triangle. So I need a lot more data. So how often do I want to monitoring? How often do I want to correlate my data are all very important sampling considerations. And a lot of people forget about the stakeholders. There may be people that are funding this research. Maybe you're working in a community that says, hey, look, we want to study this from a governmental standpoint of view. So funding is going to come down, and they might prescribe exactly how to collect your samples and what you're going to do with the data and how that's going to be used and how often as all part of your funding. So let's talk a little bit about the testing itself. So whenever you start to do this, like any type of sampling activity, I very much encourage people to start talking to the laboratory to get a handle on it. So for the most part, this is a very unique test in a sense that I don't need gallons and gallons and gallons of water. Most laboratories look for about 100 milliliters to 500 milliliters of sample, surprisingly small amount, which is again why you want to consider how you're going to composite or if you're going to do a grab sample, because essentially what we're going to do is concentrate and amplify this stuff. Next thing important to talk to your laboratory, are your goals achievable? Your detection limits. Detection limits are very important, because it's like if you've only have one or two people in the system, if you're not monitoring at the right times, you may not see that. Matrix issues can be important depending upon where you collect that at. If your matrix is dirty, there might have to be some additional cleanup procedures necessary so we can isolate the material, and obviously turnaround time. Now, PCR for the most part is a test. If it gets back in the laboratory, it can be done as quick as like 24 hours. I think probably an average lead time that you might see in a market might be about 3 to 4 days on a lot of this stuff. And it really depends upon, any laboratory's given capacity. So whenever you work with a laboratory, make sure that you can hit your turnaround time and your needs, because turnaround time can cost you money. If you want it bad, you'll get it bad, but you're going to pay for that for sure. And certainly there's data quality considerations. You need to give your laboratory the proper time to make sure that the data that they provide you has the good quality assurance, quality control, calibration, standardizations, reference materials, etc. And sometimes whenever you're doing something quick like a screen, some of those corners can get cut. So again, your data quality is very important. What are you trying to do with it, and where it's going to go and how it's going to be used? And obviously, I encourage you to contact your laboratory to get your bottles, your supplies. They're going to give you containers that are going to meet your needs with regard to volume, sterility, preservatives. Make sure you got your field supplies. Consider your shipping and your sampling instructions. Very important to collect this stuff right, preserve it right. And that goes with almost any analytical sample that you're going to be taking. So now we jump into the field time finally. So now let's say we're going to go out into the field. Again, I always encourage people to make sure you got everything. I got drivers here that'll go out and sometimes they forget the bottles. They forget their instructions. They might forget a baler. So if you're going to go out and sample and it's the time to do that, make sure you have everything ready to go. You understand what you're going to be doing. Obviously plan for field conditions. If you go out somewhere and you need to pull a manhole, make sure you got a manhole cover wrench so you can get into the manhole, open it up, collect your samples. Do I need a ladder to get down? Do I need, cones so that nobody runs over me in the middle of the street? All those things are very important to plan for. Again, I can't emphasize this enough, is how to collect representative samples, purging the locations, make sure you have clean lines, you don't have dead spots. I mean, we are looking at very, very small traces of stuff here. And as we saw before, one or two copies of this stuff can very much get amplified pretty quickly and cause false positives if somebody didn't clean the tubing on a sampler or use contaminated material. So be very, very careful that whenever you collect a sample, it's what you want and not of yesterday or contamination. And then the composite sample considerations. When do I want to collect the composite sample if I'm using the composite sample technique? Do I want a 1 hour? Do I want a flow weighted average? Do I want to collect across a 24-hour period? And all that can give you very different information, because let's say if you only run your sampler for an hour, you may not see something coming through the system, versus a 24-hour composite is going to give you much more representative sample. Obviously, fill your containers and definitely follow the sampling instructions. There's so many horror stories. I could probably do another webinar on all the things that I've seen customers do, as well as our own field guys every once in a while, with regarding to coming back with a half full container. They don't follow sampling instructions. They dump preservatives. They don't put the stuff on ice, et cetera, and all that can impact your data. And if you're going to pay for it, please get it right. Last and finally are holding time considerations. Most laboratories would like you to collect this and get it to the lab ASAP. You are dealing with genetic material here. You want to preserve it, maintain it. Most of the time that's going to be put on ice. And I do believe most laboratories would like to see that sample within 48 hours to 72 hours after sample collection. So again, some of the things to consider: are you going to drive it to your lab? You're going to ship it? You're going to have your samples picked up. But make sure you know how it's going to get there. Especially right now, if you're shipping around the holiday times, Mr. FedEx is having trouble getting packages delivered for Christmas. You don't want your samples to be lost. In the laboratory, I give you a little bit of oversight of what happens actually to the sample in a laboratory. And the first thing that happens is the concentration and purification of the sample, commonly done by filtration for this application of the technology. A centrifuge where I spin things off and decant things I don't need and keep the goodies, if you will, at the bottom of the tube. There might be other specific cleanup necessary. Let's say, you're sampling from an influent of a plant, and might have a lot of like what I'll call like suspended solids, if you will, in a wastewater sample. We won't go into any more detail than that, but that might need to be removed from the sample so that we just have an isolation of the material of interest. Commonly then, what the next step is the extraction of genetic materials. There's a lot of commercial kits out there that, if you will break down the RNA and the DNA to expose it so that it can be replicated. And again, very common genetic technologies have been used for years. In the laboratory, we certainly want to do some sort of quality control to make sure that the data that we provide meets certain standards. There's positive and negative controls. We want to make sure the machine works. We want to make sure that all of our primers, our polymerase, and all of our other goodies that we put into this stuff is going to actually do the job. We're going to make sure our chemicals aren't expired. We'll have standards, known quantities that we'll run so that we can compare our unknowns against. And very important also, that is a common thing, is the matrix spike, where I'll put in a surrogate of some sort, maybe a surrogate piece of material at the beginning of the process, knowing that it's there, knowing the concentrations is there, and then running the process to see how that's going to come out on the tail end. And hopefully I get good recovery. If I get no recovery, I wonder if the machine was plugged in. So obviously then we'll add the ingredients to copy. Those are the primers, the nucleotides, the buffers, the genetic material. In this case, it's the sample that we're actually looking for. Maybe it has some, maybe it doesn't. We don't know. We don't know if there's anything in there to replicate. And the polymerase, which is essentially the basic ingredients that help this thing kind of happen. Then what you do is you run it through your PCR and you start generating your cycles. Most labs have defined protocols to run 20, 30, 40 cycles. And again, this can be done commonly. A lot of cycles depend upon the thermo settings and breakings, and people optimize these things, a little bit different technologies, different laboratories. Maybe a cycle might only last about 5 to 10 minutes. So you can get a lot of these cycles done in a 12 to 24-hour period. And then finally, whenever we're done, we can look at those curves that we looked at before, and we can quantitate our results, and then we report them as copies of the genetic material per milliliter of sample. So now we got some data. Now what are we going to do with it? The biggest part of this whole problem is, now what do you do? Well, one of the most important things like we're looking at is the identification of the SARS-CoV-2, the specific genetic in communities and selected population. And it really allows for us to have a presence absence and notify people that, hey, we've got something going on. If you remember back to my curve of infection, your body might start putting out copies of this in the early stages of you actually being exposed to the virus, or during the incubation period, if you will, where you're really not even getting sick yet. And this is good advance notice, because most of the time people don't really go to the doctor until they start getting that headache, the cough, the fever. And at that point, the virus is already moving along pretty well. Do you want to share your data? Maybe you do, maybe you don't. Obviously, I think it's good to share the data with some people. The governments need to know that, health officials. If you start noticing or seeing outbreaks and whenever you're monitoring, well, that allows resources to be moved into these areas. You can make adjustments. You can increase your quarantine. Do you want to let this out to your community, working with your community members? Do you really want to let the news media know, that you might have a massive infection coming on? And these are all considerations that one needs to worry about whenever they collect data. Tracking is a very important part of this, very important application of this technology, because if I just do a general sampling plan where I'm looking at, let's say, the headworks of a plant, and I start seeing some positive concentrations, now I've got to be a little bit concerned of like where this is at. So now I might want to do some more sampling by going upstream, looking at things like the college, the schools, the senior citizens' homes, because or health workers or hospitals, because these are areas of concern. And right now, certainly the old folks' home is we know that's a very vulnerable population. So the ability to detect that early in that population before we've had a massive outbreak and everybody's in the hospital is, again, very valuable. Obviously, once we start getting data, one piece of data doesn't really tell a story, but as we get a lot more of it, we can certainly start to look at trends and occurrence, and we can start correlating cause and effect. Because there are some pros and cons, if you will, with these technologies, and a lot of people are still trying to figure a lot of that stuff out right now. And that's the important part of any piece of data, is you have to understand the limitations associated with that so that it can be used properly. So finally, we look at some pros and cons on this. Is, like I mentioned before, I think it's a very, very good tool to where we can look at presence and absence. Hard to correlate it right now, maybe with the concentrations, because we don't know where people are at in that infection cycle. Have we missed it? Are we on the increase? And again, you need multiple data to do that, but definitely a tool that we can use. Again, using the proper application of the response and resources. If I have this stuff spotted, who do I call? What do I do? Is it a lot? Am I on the up curve? Am I on the down curve? But certainly we have to understand that and apply it accordingly, because if we call out the dogs and we're on the tail end of the infection, or we missed it, or we've got changes in concentrations in wastewater because of changes in the demand in the system, all those things need to be adjusted for, because we could only have a few people in the population, but we've got so much dilution that we just don't see it, or we didn't sample at the right time. Then we could see a lot of it coming through because maybe there's a lot of retention time in the system, and we missed the wave because it happened 5 days ago, because it took 5 days for the water to ultimately get to the plant and sampled. Good thing about this is it's definitely a quick result, and it's a tested technology used in many hospitals, many researchers. DNA, RNA studies have been done for quite some time. All this technology has been done is adapted specifically for the SARS-CoV-2 virus. Again, a little bit of a con, understanding the data, concentration versus occurrence, the standardization of sampling and methods right now to obtain comparability. And comparability is a very important parameter in any type of laboratory you work, because certified labs, accredited labs, we all want to make sure that we're doing the procedure as written, so that if you come here, if you come there, the method should give you the same result. So right now, there's a lot of people jumping into this, and there's a big effort right now to standardize the PCRs, the primers, the cycles, how you sample, so that whenever I get one piece of data and I'm looking at another researcher's pieces of data, that those two can be compared equally without any method bias. Obviously, it's another tool in the fight, but it's used to complement other things. We certainly can't give up on the other things that we do, the clinical testing, the hospital testing, all across the place. But it is another tool for us to put in the effort. And again, finally, the data is really only as good as a sample. I often look at here at the laboratory that we work at here in ALS Middletown. I think we do a wonderful job. We have a lot of quality control. We're audited. We all have different type of accreditations. But you know what? That's only as good as the guy in the field that sends me the sample in the container. Is it representative? Was it collected right? Was it purged right? So definitely we got to make sure that we standardize some of that. So a little bit of a conclusion here. Using PCR to detect the SARS-CoV-2 in wastewater is a cutting edge way to evaluate virus presence in communities in a non-intrusive and especially in non-clinical setting. There's still a lot of variables because this is a new technology. We need to standardize that with sampling and analytical protocols. And indeed, we need to collect more data so that we can evaluate that and we can interpret the test results properly so that we can either control or contain outbreaks. But with that said, the more tools and knowledge that we have, the quicker we can beat this virus and get back to our normal lives. That pretty much concludes my presentation today. So like with that said, obviously I'd like to give a big thank you for you spending the last, looks like 45 minutes with me, listening to me talk today. Hopefully you got a little something out of this. Again, I'm Ray Martrano. I'm our operations manager here at ALS Middletown, 301 Fully Mill Road. And that's my contact information. So I'll turn that back to Alex now. Thank you.
Thanks, Ray. We're going to open up the form for some questions now. We already have a couple that were submitted. And I'll give you guys a little bit more time to submit questions as well. First of all, we'll ask, we have a question from Cynthia asking, what is typical cycle number, and when are more cycles decided to be done?
From what I've seen, I mean, usually about 40 cycles is common because you really start to amplify this quite quickly. You know, and again, that's a little bit based upon what you want to do as far as an objective. What do you want to call non-detect? What do you want to call detect? How long do you want to run this thing? But you also have to look at like a time involved with that. So again, that's where we need a little bit of method standardization. But I would say generally anywhere between 30 to 40 cycles is most common what I've seen with that. Okay.
Okay. We have a question from Sudhir who asks, what is the cost for each sample for normal turnaround and the cost for rush samples?
As like anything with a laboratory, I always tell people to consult the laboratory for that, because laboratory pricing a lot of times is not necessarily like a commodity pricing. Laboratories are going to price the work based upon a couple different things. Number one is, can I even achieve what you want me to do? How much do I need to knock off the bench and put your sample first? And the other thing is, how many of them are you actually going to give me? Because then I can isolate time. I can have efficiencies in processing. Like anything, it's very hard to do one of something, and it's a lot easier to do more. But typically, market price, and I'm going to take a guess at it, is anywhere between about 75 to about 125 a sample. And if you start looking at rush turnaround times, that can easily double or triple the sample time. And again, it depends. If you want something done on a Saturday and a lab's got to bring somebody in on a Saturday to set the sample up and run the stuff. But there is definitely cost associated with the primers to do this. But that would be a fair estimate, I believe, of where the current market price is.
Cool. We also have a question from Melinda. She asks, would purification or filtration affect sample concentration?
That's where you really can look at the importance of putting in a surrogate into that. Just like any analytical protocol, you know, I always look at like DEP, who's come out with all these wonderful methods, you know, and they sit somewhere and they're doing like clean drinking water and stuff, works very good. Whenever you get into matrix effects or purification, that's where the surrogates or the host bacteria that you might put in there. I've seen people using like a bovine kind of in there. And let's say if I come back and I get maybe a 60% recovery on that matrix spike, well, that's going to tell me that for whatever reason, the matrix was repressed. And myself as the end user, then if I would look and say, hey, I got a 60% recovery, then I probably need to multiply my data by an additional 40%. Laboratories will not surrogate correct things. Typically what will happen is they will have control ranges on their matrix so they know they're doing it. But it's really hard to say because a lot of different matrices will affect the sample differently. And hopefully the laboratory, the filtration process or the centrifuge process, some laboratories will have the flexibility to say, hey, look, this is not going to filter well. It's going to be better if it's centrifuged first, then they might centrifuge it and then filter it. So a very difficult question to answer, but that is why you're using some sort of a matrix surrogate in there to give you an indication of how good the recovery actually is.
All right. Thanks. Just to reiterate, if anybody else has any questions, you can use the Q&A function in the toolbar at the bottom of the webinar. And attendees can also see all submitted questions in that window as well. I'm going to give a couple more minutes to let everybody else type in the additional questions they have, and I'll just go over some brief notes. This webinar presentation has been recorded. All participants of this webinar will receive a follow-up email once the recording is available to view. We also post our webinars on the ALS Global website and our ALS YouTube channel in the webinar playlist. I encourage you to find a wide range of video resources and past Webinar Wednesday presentations available there. All right, we have another question from Melinda. Are there QC samples associated with this sample? Temperature blank, field blanks, etc.
Yeah, and now as far as the field goes, it sounds like that's more related to field type applications. Again, that's where you go back and you actually look at like what your sampling protocol is. And through the years, I've learned that, but what's the best way to put this? You know, depending upon the data quality objectives, meaning, am I looking to tell the truck or the backhoe to keep digging because I've got a contaminated spot, or am I going to do some sort of a closure sample where it's going to go to court? It's going to have very high visibility. Well, in the first thing, you know, I just need some quick and dirty stuff. I don't necessarily need temperature blanks, equipment blanks, a lot of quality control, matrix spike samples, etc. I just need to know, like, do I need to keep digging to get rid of the contaminated, maybe petroleum spill, for example? If I'm going to try to permit an incinerator next to a kindergarten class that's burning PCBs, well, you darn well know I'm going to have equipment blanks, temperature blanks, QA, QC, and that data is going to be bulletproof because I know I'm going to be in court and it's going to be challenged. So that's really up to the end user on how much field quality control they're going to do with that. In the laboratory, under the basic methodologies, there will be positive, negative controls. There will be spikes, if you will. There'll be controls testing on the media, the matrix. But from a field perspective with regard to temperature blanks, trip blanks, etc., that's almost up to the end user then. But be mindful that that will run up your cost. So in my simple example, if I'm just kind of digging the backhoe, I really want to just do something quick and dirty, almost like a screen, because I don't need a high quality piece of data. I'm screening, in this case, maybe for SARS, I'm just looking for presence absence. If I find it, now maybe I want to spend more money, more field time to get better data quality that is quote unquote more defensible.
All right, thanks. It doesn't look like we have any more questions. Let me just double check. All right.
Yeah, you have my contact information too, anybody. You're welcome to send
Yes.
me an email or phone call.
Yes, correct. So in our follow-up email, it will have Ray's contact info and the recording. So you can send any questions that come up afterwards directly to him. I just want to thank everybody for attending today. I hope you guys enjoy the rest of your week. Thank you.
Thanks, everybody.